Hello, everyone. See people starting to pop in. Or just give a little time for everyone to get joined here. Okay, it's 5 o'clock right now, but we're just gonna wait a couple minutes for anyone else who wants to log in. Welcome everyone. Righty. Hello everyone, and welcome to Café Scientifique from all of us at SFU's Faculty of Science. Um, We're going to get started here. So if you're one of our cafe regulars, I just want to say thank you for joining again. And if you're new, welcome. Thanks for joining us for the first time. Great to see some familiar names and some new names. I know we have guests from all over British Columbia and outside. Uh, my name is Stevie Benish. I'm the acting manager of outreach and engagement in the Faculty of Science. And Café Scientifique is the Faculty of Science public seminar series where our faculty presenters share their current scientific research and its impact for our local community and for larger society. So today we have our second cafe of the academic year, and we're looking forward to the continued talks we're having throughout the rest of the fall and into the spring. We typically have about 6 to 8 discussions a year, and they're all virtual. They're also recorded, so if you missed something during this talk or you would like to share this presentation later with friends or family, it will be posted on our website later. And we'd also like to hear from you and stay in touch after the talk, so you will be receiving a feedback link by email, and if you're interested, you can join our mailing list and receive updates about our upcoming events. So tonight, I'd just like to start us off on a good way by acknowledging that we here at SFU Burnaby are privileged to gather today on the unceded and ancestral lands of the Musqueam, Squamish, Tsleil-Waututh, and Coquitlam peoples. We thank them for having cared for and being stewards of these lands and waters since long before our time. We look forward to working with them in partnership as we continue to build and grow our communities together. If you know whose territory you are currently on, I invite you to take a moment to reflect on this. And if you don't know whose territory you're on, a great resource for more information is the website native-land.ca. And there you can familiarize yourself with the Indigenous people whose land you both live and work on. Okay, before we get to the talk, we just have a couple little house rules. So first off, If you have any questions throughout the talk, we will have a Q&A section at the end, so feel free at any time to type your questions in that Q&A box, and then we'll get to them during our Q&A section at the end. If we have a ton of questions, which hopefully we do, but if we do have too many and we don't get to them all during the presentation, we will follow up afterwards via email. And so on that note, you will see the Q&A box, but the chat function is turned off. So once again, just use that Q&A for any questions. Just other details are the live transcript is on, so please feel free to use that. It's for your convenience. And just another note that this is being recorded and it will be shared kind of on our website as well as our YouTube list and all that. Alrighty, so let's get to tonight's discussion. I would love to be able to introduce our speaker tonight, Dr. John Reynolds. There he is. So I'm very pleased to be able to introduce Dr. John Reynolds. He's a professor in the Department of Biology. John holds the Tom Buell, I've definitely said that wrong, Tom Buell BC Leadership Chair in Aquatic Conservation, and he recently finished a 4-year term as chair of the Committee on the Status of Endangered Wildlife in Canada. He has published 5 books on conservation and ecology and is a fellow of the Royal Society of Canada. His research at SFU focuses on the conservation and ecology of Pacific salmon, with an emphasis on the wider role of salmon in freshwater and terrestrial ecosystems. So tonight, Dr. Reynolds is going to talk about his research, including how salmon act as kind of a conveyor belt for the nutrients moving from the sea to streams and land in BC's coastal rainforests. We're very excited to have you with us tonight, and I'll pass it over to you, John.
Well, thank you very much, Stevie, and really appreciate the opportunity to give this talk, and also I appreciate the interest that people in the audience are showing in this work. And I have a similar land acknowledgement to the one you've just heard from Stevie about the Burnaby campus. I'm on the lower mainland of Vancouver on the territories of Coast Salish people, and I'll also be introducing you to the territory of the Heiltsuk First Nation, where I've been doing the work that I'm about to describe to you. So I'm going to share the screen and we can get straight into it. Is that looking good, Stevie? If not, you can just let me know. I assume so.
That looks great, John.
Okay, thank you very much. So yes, marine, basically marine fueled ecosystems in the Great Bear Rainforest have been an interest of mine for about the past 15 years. Ever since I joined SFU, we have been working on this, and I'm going to be telling you about the work that I've overseen, but of course the real heavy lifting has been by armies of graduate students, undergraduates, postdocs, members of the Heiltsuk community who we have collaborated with as well. So it's going to be a bit of a flyby through some of our highlights of the work, and I am certainly keen to discuss anything further with you at the end of this talk, as Stevie mentioned. So as an ecologist, I tend to be often getting somewhat focused on particular habitats. Ecologists can't study everything, and most of us tend to identify either with the marine environment, so which is what is in the estuary that I'm showing you here, or you could look further upstream at the freshwater environment, or you could look at the mountains and study terrestrial habitats. And by and large, we often don't need to be experts in all 3 ecosystems, and depending on the question, there are certain questions where that's okay. We can go to different conferences. The marine people have their conferences, the freshwater people have their conferences, and so on. But there are some species which make a bit of a mockery of our attempts to silo the habitats in that manner. And of course, the most obvious one that you know I'm going to talk about is salmon, because Pacific salmon die after spawning, and they get eaten by an awful lot of predators. And because they are born in freshwater, they then go to the ocean, and while they're in the ocean, they put on most of their weight. So they're growing at sea. It's really very much a marine fish. But then they come back to freshwater again to spawn, of course. And when they do that, then they can die after spawning. They can be eaten by bears or wolves or other predators, and their nutrients can end up on land. So they can tie these 3 different habitats together. And that's what I've been very interested in studying for the past number of years. And so what does it look like? Well, On the top left here, that's a chum salmon, and that's my boot for scale. And this is a fish that's been killed by a wolf. Or you can get accumulations of carcasses after spawning when they die naturally, as they're programmed to do after they run out of energy at the end of their spawning season. And that could be what you're seeing on the other side of the screen. And so right now we are in peak migration, peak spawning for many species of salmon in many parts of British Columbia. And they do, as Stevie mentioned in her intro, you could think of them as conveyor belts of nutrients because they are acquiring these nutrients at sea. They come back and they leave them behind one way or another. And these are often going into ecosystems that are somewhat impoverished for nutrients. So the nitrogen in particular for the terrestrial ecosystem might be having an impact. We use nitrogen to fertilize our plants and our crops. Well, is there a fertilizer effect happening with salmon? That's one of the big questions that we have been interested in on trying to address. And so salmon potentially are tying together these three different habitats, and I'm going to be focusing a little, well, actually on all three, but going to be quite a bit of time mostly on the terrestrial side and somewhat on the freshwater as well. And I'll be doing this by giving you a few examples of the work we've been doing, and at the end I'm going to come up with a wider view of things, including a review that we've done, which tries to see how widespread the effects are that we've been looking at. One thing that might surprise you about Pacific salmon is that they have an enormous distribution. The different species can go from Baja, California, all the way down to Japan and Korea, and so they actually are covering a very large area. Now, of course, not all of Alaska is green with these salmon. This just reflects the overall distribution of how far inland they can penetrate on different river systems. So there is a potential that if they're having a nutrient effect of some sort on terrestrial ecosystems, this might actually be covering a fairly large region. It's not just a local British Columbia phenomenon, for example. If you do what I did when I first was interested in getting into this, and you Google salmon carcass, you might come up with something like this. This is a photo from a little guidebook to how to put carcasses into streams. Now that might be something you're going to stop and think, what on earth does that mean? Well, you see the person in here is wearing gloves because that's a frozen fish. This is a hatchery, and the truck is full of carcasses of salmon because when hatcheries strip the gametes from the fish, they kill them. So they have these carcasses left over. What are you going to do with a carcass? Well, there are guidelines suggesting how you should put them back into streams because that, so the theory goes, might then have a beneficial effect on the stream system by fertilizing the stream, maybe even helping to then feed up through the food web and lead to more salmon being produced from a highly productive stream. That idea has taken hold. It's quite popular in many places, but before we get too carried away, we need to think about one other effect that salmon have, and you can see it quite easily. If I, let's see if I've got my volume on high. It doesn't matter if you can't hear this one. I'm not sure if there's a volume, but this is a video I took in our study region. Oops, let's go back a second. These are chum salmon. We've got a couple males fighting it out, waiting for a female to spawn. Now, if we think about what's going on there, the general mayhem, of course. The females are digging their nests in the gravel. The males are jockeying for position, fighting with each other. All of this is stirring up the gravel. And one of the effects it can have is it can actually remove nutrients from the stream because in the form of the insects that live in the rocks, which have been shown to get swept downstream and out of the system. And so there actually could be suspending a lot of organic matter, including living insects, and removing them from the system. So it's not necessarily a complete positive effect that this so-called conveyor belt is having. There can also be a negative effect on certain components of the stream, which would then potentially even reduce the nutrients, as some people have argued. So when I began this work, it was a little bit of a kind of a toss up which way this was all going to go. I was definitely not the first to start studying these questions, but I was very keen to find a place where we could study a large number of streams and try to get a handle on this. And again, I should just mention this physical effect here. These, it looks like snow drifts in the stream here. These are have been piled up by spawning Chinook salmon. They move these big rocks. It's a big fish, and they go downstream and they land, and it looks like waves of debris here, of gravel in the stream. So this engineering effect should not be underestimated. So I set up, I was very lucky to be able to set up some work in the Great Bear Rainforest. This map shows logging in British Columbia since 1970. Brown means logged. And you can see there's this area in the central coast of British Columbia where there has not been very much logging. And this is the area that is referred to as the Great Bear Rainforest. It's hard to get there. It's extremely remote. There's lots and lots of islands, and so it's difficult for these sorts of industrial activities to have occurred in any large amount. And I was very keen on that because I wanted to try to study a large number of streams that have not suffered a lot of heavy industrial impacts. So I was very fortunate to be welcomed by members of the Heltzik community who are based in Bella Bella here. And so I've been working on I'm on a different island from theirs, but just 15-minute boat ride away. And I have a base there year round which we use as our field base. And I've been working with people from the nation in various capacities, especially fisheries biologists who are based there. And together we've been counting the fish every fall, which is a major activity, and also all of our data are shared with the nation, and we try to help out in other ways as we can. So this is what a day in the office looks like for me when I'm lucky enough to be there. And so this is one of the fjords where we are working. You have to share your office with others, of course, up in this beautiful region. It's not always sunny, and this year it was so rainy that we actually had to curtail our salmon counting efforts early because we couldn't. The streams were so high and so violent that we couldn't safely work in them, and we couldn't see the fish. But on other days, it can look like this. So this is an example of one of the larger streams that we're studying. We also study much smaller streams like this. So they can vary greatly in size. And as I said, these have not really, they've certainly been modified culturally over millennia by the Heltzik people who live here, but they have not been harmed in the way that happens with, say, forestry or urbanization or pollution. They're in very, very good shape. So here's an example of salmon counting. When we did this work, we didn't know how many fish were in a lot of the streams, especially the small ones, because nobody was counting the fish. So here we are. I'll just play a little clip of us. These are mostly pink salmon that you're going to see in this one. You can hear the ravens. So my lead salmon counter, who kind of directs traffic and the operation for many years, has been Howard Humchit, a good friend in the village. And that noise, I don't know if you could hear it, somebody was yelling, "Hey, oh," the sort of the standard way of letting the bears know that you're coming. Oops, we'll move on. So I've already shown you briefly what this looks like. This is, as I said, a very typical wolf kill. They often don't eat much of the musculature at all. And this is sitting there, and my question is, well, what's it doing, if anything, to the biodiversity of the area? So this map shows all the different streams that we have been working on, along with people from the Department of Fisheries and Oceans in collaborative counting, as well as the Heltzik First Nations. All of our locations are reached only by boat. There are no roads, leading to any of these streams on these. They're mostly on islands, and so this is all boating. And we work within about 2 hours of our base. Our base is right dead center in the map there, opposite Bella Bella, and we go up to we can get to about 2 hours away is workable. Now, the main stars of this particular story are both bear species, grizzly and especially black bear. And then we have these fishing wolves, and they really do. They're not just scavenging. Believe me, they kill a lot of fish, especially pink salmon, but chum too, which is quite something because a chum is a heavy fish. And so we see wolves quite frequently, actually, and we certainly see the results of their work, which can be hundreds of carcasses spewed along streams which have been hardly touched. So we've been looking at many different aspects of this system. Don't worry, I won't show you them all to you, but for example, if you want to think of this as a bit of a food web, if you have a pink salmon at the bottom, we've looked at the effects on stream algae, we've looked at effects on stream invertebrates, aquatic insects. We've looked at coho, I'll tell you about that one, and we've done a lot of work on sculpin as well. But on that axis, I'm just going to mention the fish one. And then we've also worked on bears, a lot of work on plants. I will tell you about the plant work, and I'm going to tell you about the bird work as well. So that's where we're going to go. And the first place I want to go is with the fish one. I mentioned that there was this idea that if you put the carcasses into the stream, maybe that can help to the next generation of fish because it will cause more, there'll be more food for invertebrates, for example, and the fish then can eat those invertebrates and grow faster and so on. That was an idea which had really no support when we began our work. So we wanted to ask about this. And an important part of the biology of the pink and chum salmon that we study is that this is a chum. They do not feed in fresh water. So the adults, of course, all salmon stop feeding when they come back in the fall. But even the young ones, when they come out of the gravel where they were in their nests, when they come out in the spring, they go straight to the ocean. They do not feed. So it's not possible that one generation of chum salmon is helping the next generation of chum salmon because they don't eat. So they couldn't benefit from a more nutrified stream. But coho is a different story. So the coho up in the top right there, those fish spend 1 to 2 years as juveniles feeding in the streams. And so we wondered whether there could be a crossover effect from pink and chum salmon to coho. And I'm going to make a very long story short by showing you one key graph from the PhD by Michelle Nelson. And on the bottom axis, you see the number of spawning pink salmon in each stream. So each dot is one stream. Okay, so the streams go from no salmon, because there was a waterfall blocking them, all the way up to maybe 6,000 pinks. And on the y-axis, we have the juvenile coho abundance. And you can see that at low numbers of pinks, the abundance is quite variable, but it's uniformly high when there are a lot of pink salmon there. Now, this is a correlational study, and you always have to worry that maybe a stream that's good for pinks is good for coho. We tested, in fact, 17 extra variables here that might explain this result, and we competed them against each other and against pink and chum salmon. And the winner, every time we did this, ran this little competition saying which one has the highest explanatory power, the winner was pink salmon. Pink salmon was the most important factor that we could find that appears to influence the coho abundance. It also they're also related to their growth rate. The coho grow faster in these streams as well. This may be partly because they can be predators of eggs, of salmon eggs. So they may be eating eggs of the pink and chum salmon and also the juvenile pink and chum salmon when they come out of the gravel. So it could be a direct predation effect as well as possibly through enhanced invertebrate abundance, but we're not sure about that. So that's all I'm going to mention for now about the fish effect. Instead, I want to move into plants because we have spent a lot of time looking at plant diversity, and I wanted to know whether the plants that grow beside the streams could be affected by this nutrient fertilization in the form of nitrogen. And so let me introduce you to one of the craziest experiments that I have ever done. And I want to give credit to Morgan Hocking, my former postdoc, who led this one and did the work with me on this. This is a waterfall, as you can see, and this waterfall blocks the salmon. They cannot get up that waterfall, and there's no habitat. The ocean is basically more or less where I'm standing. I think I was in a boat when I took in the boat when I took the photo. So there's no salmon in this stream. Okay, think of it as a control. Well, Morgan and I, one day went to a stream nearby, and we got salmon carcasses, one fish that had already died. We put them in plastic bags. We boated them over to this stream, and then we walked up. We bushwhacked on a hot day up past the waterfall, and we set them down to do a little experiment. And chum salmon are big, as I mentioned. And I remember at one point we were it was really tough work. I remember catching my breath with two dripping bags of rotten salmon. And Morgan turned to me and he said, "I hope we don't win a Darwin Award for this," because we were carrying salmon in grizzly bear country. And fortunately, we both survived this particular experiment. But why would we do this? Well, here was the idea. You go in the fall and you put down a rotten carcass. We went for rotten carcasses because we were hoping the bears wouldn't scavenge them. So you take really rotten carcasses and you set it down on the ground, flag it, and then we took a sample of plants from near the carcass and away from the carcass. And far means meter and a half, maybe a meter away from the fish. So you take plants near where you're about to put the carcass down and away from the carcass. Then we returned in the spring and we did it again, the plant sampling, near and far. And by this time, the carcass was a skeleton, and most of the carcasses did stay there, so it worked. And so we sampled the plants before and after we put them down, and near and away from the carcass, to ask how the presence of a carcass might affect the physiology of the plants. And we did this on a whole lot of streams, by the way. We didn't just do it on this one. We did it on a wide range of streams. So here's the result. On the y-axis, you have the percent nitrogen in the plants. Okay, and these are just three that I'm showing you. We also studied one more, but these are just foam flower and bunchberry and false lily of the valley. And then the different treatments are shown, the 4 different treatments. So in the fall, at the carcass, you had the same percent nitrogen as the sample away from the carcass, which is a good thing because we hadn't put the carcass down yet. They should be the same. Okay, so that's fine. Near and away, no change. We hadn't done anything yet. But if you go back in the spring, you can see a bounce in the gray box there. There's a bounce upward in the percent nitrogen in the spring at the carcass. And it's higher than away from the carcass, although away from the carcass has a little bit of an increase too, simply because spring plants have got more going on in their leaves. So this is the first experimental demonstration that carcasses can actually affect the percent nitrogen in these plants. And in fact, the fewer salmon that were in the stream, the bigger the effect, which makes sense because in the streams that where we did this, where there were a whole lot of salmon, they probably already were getting toward a saturation level, and our addition of a carcass didn't make very much of a difference. It made the biggest difference when there were the fewest in the streams where there were the fewest carcasses. So that was fine. We thought, okay, we're getting somewhere with at least at the physiological level, but it's a long way to go from there to saying it actually has an effect on the ecosystem, even on the community of plants. And so at the same time we were doing this experiment, we did a fairly ambitious study of 50 streams. We went to 50 separate streams and we sampled the plant communities. So here's a quadrat, and we looked at the percent cover of everything from the moss layer up to the shrubs, and we ran a transect. We did 5 of these on each of 50 streams. You can see a transect tape there in the trees. We measured the diameter of over 6,000 trees along these transects. And so it was a lot of work. That's Morgan Hocking there, who deserves the credit for leading this study. a heck of a lot of work by a team of dedicated people, and I have somewhat fond memories scrambling up muddy banks in the rain, bushwhacking up with the transect tape, laying out the transect, then throwing these quadrats at regular intervals along it. So we did all of that, and let me just introduce you then to some of the results. I'm going to take you through this sort of one step at a time. So you can see we've got 4 plants across the top. These are just representative plants. Um, and the two on the left, the blueberry and azalea, are plants that don't really need nitrogen. They can access nitrogen from other sources like rotting wood. The two on the right do. And so salmonberry is an example. They can benefit from the form of nitrogen that carcasses provide. And so each dot is a stream. Okay, each dot. So again, I can't sort of, maybe I'm thinking of nightmares rather than happy thoughts here, but each of those is multiple transects, rain or shine, up the stream, multiple quadrats per transect. And so we've got 50 data points here. And so you see the first column row here. This is the heavy nitrogen in the plant tissues in the leaves. Heavy nitrogen is the isotope of nitrogen that can indicate a marine origin. Marine things have higher level of heavy nitrogen than freshwater things, and it also amplifies so up the trophic level. So if you have a high level predator, for example, which salmon are, then you're more likely to have more of that heavy nitrogen. And the interesting thing is that in all 4 of these species, it's a almost identical relationship with salmon. The more salmon that are in the stream, the more heavy nitrogen that you have in the leaves. And so that fits with the idea that the salmon might be a source of some of this nitrogen in these leaves. But it doesn't matter whether it's a species that we already knew could benefit from nutrients because of its physiology, like the salmonberry, or something like a blueberry. So then let's look at percent nitrogen. Remember, we did that with the experiment. Here's the percent nitrogen. In the 2 on the left, these are species, remember, they don't really benefit from salmon. They can live on rotting wood, whereas the other 2 cannot. And there's no change in percent nitrogen. So they may be taking up nitrogen from a salmon origin, but they're not. They're not better off in terms of percent nitrogen. Whereas the 2 nutrient-rich indicator species on the right, they are, in fact, have more nitrogen. So this is a correlational analog of the experiment I just showed you. Now we've got 50 streams asking what happens in the real world. And then if you look at the relative densities of these plants, though, from our quadrats, you can see that the 2 that are not particularly nutrient-rich indicators, in fact, go down in streams with more salmon. One of them has no effect, and then the salmonberry actually goes up. So we have more salmonberry on streams that have more salmon. Okay, it's a well-named shrub. I must interject at this point and say that when I've spoken to my friends in Bella Bella about this, they weren't surprised at all. They were quite aware of the fact that there can be relationships between salmon and salmonberry, and historically people have fertilized salmonberry plants by sticking carcasses beside them. So I may be a few thousand years late to understanding this, but this is the first time anybody has quantified it in this manner and shown what the relationship looks like. You can also group all of the plants together. So I showed you 4 examples. In fact, there's a lot more than that we studied, and you can bundle them together into our nitrogen-rich community indicators and nitrogen-poor indicators. And if you do that on a scale from a whole bunch of species that really don't need the extra nitrogen to the ones that do, that's what we have going on in the y-axis. And you can see as the density of salmon goes up, you shift the community beside the stream toward these nitrogen lovers. So things like stink currant, false lily, certainly of the valley, the salmonberry, as I mentioned, these kinds of things really get going. Whereas things like the blueberries, deer fern, salal, things like that. They really don't need salmon, and those tend to be um, more common in the streams that don't have as many salmon. So it's the first time people have shown this sort of shift in terrestrial plants. I should say this is all within about, I think our transects were about 35 meters long. So I'm not saying this effect goes much further than that. In fact, we have some evidence it probably does not. But it's a stream side effect. But these are not aquatic plants. These are terrestrial plants. So one of the things that I think is really important, actually, let me go back a second. If you look at this graph with a critical eye, you could say, well, hang on, is a lot of variation around that line. Some streams have got way more nutrient-rich indicator plants than you would think, given how many salmon there are, and some don't. So what's explaining all the scatter around the line? Well, two things explain a lot of the variation around that line. And that's what I want you to see here. Physical characteristics. So on the left, we have a steep-sided bank. We measured the gradient of the bank. And in these kinds of canyony, very damp, humid, wet places, it doesn't matter how many salmon you have, the plants beside it are not really affected. And I think it's partly because no bear in their right mind would come down, this slope and use that as a foraging platform. And even if they did, the carcasses that they leave behind are not going to stay there. They're not going to stay on these slopes. They'd wash out in the first rain. So these kinds of systems, it really doesn't matter how many salmon you have. They have no impact that we can detect on the riparian plants, the streamside plants. In contrast, the one on the right shows a lot of red alder, and red alder fixes its own nitrogen. So it can actually be a source of nitrogen to the surrounding plant communities. And there too, the number of salmon you have doesn't matter, as you might expect. We had already wondered. Fortunately, we measured this. We looked at how much alder was there because we'd thought of this beforehand. But so whether you have a lot of salmon or not, the alders, these are not nutrient limited systems. The nitrogen is already being provided from another source. So these are two exceptions that I think help prove the rule. And when these results came out, that was kind of my eureka moment, to be honest. It was then that I thought, okay, I think if we can explain when you do get an effect, and also when you do not, I thought maybe we're onto something, and that we have we really do understand better how the salmon can and cannot affect the streamside plant communities. So I think right now, if you took me to that part of the world and blindfolded me or didn't tell me how many salmon were there, I think I could look at the vegetation and at least be able to say low, medium, or high in terms of number of salmon by taking these things into account. So I'll leave the plants behind, and now I want to move on to birds. I'm a keen birder, and when we first started going there, I spent a lot of time trying to figure out how we could try to study terrestrial birds in relation to salmon. The problem we have is that a lot of the streams are quite noisy, and it's hard to do the kind of counts of birds that you do by listening to their song. We eventually overcame that, but the first study we did was by Rachel Sudbury, who did her master's with me, and she had a background which included not only birds, but also forest ecology. So she knew how to do forest plots and how to account for all kinds of aspects of the forest that might affect birds independently of salmon. And I'm just going to show you two graphs from this work. Here you see the total carcass biomass in streams. They're not all different streams. Now, in this case, we visited some of them multiple times, but we controlled for that in our stats. And then on the y-axis, you have the abundance of scavenging birds. So these include about 5 species of gulls, bald eagles, ravens, that sort of thing. And you can see that in the streams that have the most salmon, you have the highest numbers of scavengers in the estuary. I should have said this is in the estuary. So I think so. As you boat up, you don't even have to get out of the boat. You can just do this from the boat, and you can count the birds, and you get what you would expect, that streams that have more fish have more scavengers. More interesting, though, to me, is the breeding bird assemblages. So here you see the combined pink and chum biomass of different streams and the number of birds per hectare. And this is based on 2 years of data. So for example, if you go to the far right, you can see the two streams that have the most fish were visited twice, and they have similar birds per hectare. So that's why there's a pair of data points there, one above the other. Each is a different year for the same stream. And you can see then that as the total biomass of salmon increases, we get a higher density of breeding birds around the estuaries in the forests. So these are breeding birds. And one of the things, though, to think about, though, is some of these birds have never seen a salmon in their life because they're migratory. So, for example, the Townsend's warbler or western flycatcher, these birds leave before the salmon arrive in the fall. They're finished breeding and they go. Okay, so they're not eating salmon carcasses, they're not eating salmon eggs. We think what's probably happening is that the insect abundance overall around the estuaries is higher as a result of the numbers of salmon. Some of these insects feed directly on the salmon, others may be supported indirectly through the nutrients from the salmon. But that's what we think is going on. And again, I want to emphasize that Rachel used her forestry background to control for all kinds of aspects of forest structure that may or may not have been causing a spurious correlation. This result stands up even when you control whether or not you control for all of those other variables. So this is the first study to show that the breeding bird densities around salmon estuaries are higher when there are more fish. Well, we wanted to go a little further with this, and Kirsten Wilcox did some work on this, and we wanted to take one of these birds and look more deeply at what might be going on. And so the bird we chose was the Pacific wren. Many people who live, probably everybody who lives in the Pacific Northwest might be familiar with this bird. And let's see if you can hear it. I'll just let you. So that's one of the sounds of the classic sound of the coastal rainforest. So the method that Kirsten used was territory mapping. And now this is a caricature that she's drawn. She's very good with graphics. And the birds we did band the birds, color band them so we could tell who was who. But even before we started that process, you could also tell how many birds were on the territories just by going and making repeated visits and mapping where they are. The question we're asking is whether there are more Pacific wrens on streams that have more salmon, and what the consequences are for their reproduction and behavior. So this is an actual sketch of a map. So the stream is shown schematically in blue. And these circles are detections of a bird, either visual or more often you hear them because they sing a lot, they scold a lot, they kind of give you hell when you walk into their territory. They tell you exactly what they think of you. And so as you walk along, you can map them. And so what Kirsten did is we have 4 detections of one bird in one place and another detection in red of a second bird. Then you go back and you do it again. And again, we have detections in 2 different places, and even if they're not color marked, given their behavior and where they are, you can kind of get a picture of the fact that there are in fact only 2 birds in this region. So there's the map that starts to emerge from subsequent visits, and indeed 2 birds. And these are tweeting and retweeting each other, and clearly you could draw a line around now what this territory is, and you can say how big their territories are. And so that's what we did. So this one on the left, the schematic, that's that first stream I introduced you to where we brought the dripping carcasses up for our carcass experiment, Ripley, with the waterfall. No salmon there. Okay, so that's what the no salmon symbol means. And there are our 2 territories. These these males have divided the territory this way, and there's two birds. Okay. Here are four more. We've got some with more salmon and even more salmon on the right. The Klatsy, that particular river, has the most salmon of any of the ones that we study. So lots of salmon, and look at how many territories there are. Notice what they're doing. It's the same area. We studied the same total area. So what they're doing is they're packing in. They're smaller territories. They are more specific about where they put them. We mapped the trees in the territory, and they're really just packed in huge numbers. And again, I should say the Pacific wren, I've never seen them eat. I've never seen them scavenge at a carcass. I'm sure they might sometimes. They're pretty opportunistic, but they're not a. It's not a salmon predator by any means. It's a tiny little bird that eats insects and spiders and things. And so one of the things that was very noticeable was that on the streams that had very few uh, salmon, the birds didn't sing very often. They didn't have to. They know who's who, and they would sing now and then at each other or scold each other, but they kind of had it all sorted out. Whereas the ones that had a lot of salmon, it was mayhem. The birds were defending their territories much more vigorously. They had all these other neighbors to worry about, and so the males were a lot more busy, just defending their territories from all these other birds in the high density streams. But the interesting thing is, if you look at the number of young that came out of the nests from different streams, the young per nest was the same. So the productivity of an individual bird was the same, whether it was on a stream with no salmon or a stream with a lot of salmon, because they are piling into the streams with the most salmon up to the point where everybody's doing about the same as they would if they went to a stream that had fewer salmon. So there are more young birds being produced in a high density stream, but the per capita success rates of the birds is equivalent across different densities. So what we've got so far is, and we've looked at many different things. I didn't tell you about the algae, but which is the first rung up this chain of events, but definitely the algae go down when the salmon arrive. The stream invertebrates go down when the salmon arrive. I've told you a little bit about that coho fish, that we have higher densities with more of them. We've talked a lot about the plant community shifting toward nitrogen lovers and some of the behavioral ecology of the birds as well. My question then becomes more broadly, and this is going to be the final part of my talk, how common is this? Because you need to understand that there's been some healthy doses of skepticism about really whether this is a widespread phenomenon or not. We seem to find relationships everywhere we look, but not everybody does, and there has certainly been some pushback against the idea that the salmon were in fact having major impacts on terrestrial ecosystems. So Jessica Walsh joined me. She's now a professor in Australia. She joined me for a postdoc and did a quantitative review of all the research that has ever been done on this question. And so it's the green at the top. I'll just take you from the top. You can start off with, we found just by doing searches, you could find about over 3,000 potential papers that seem to fit the bill for having something to do with the effects of salmon on X. Okay, and then if you screen out all the ones that in fact are not helpful, you get down to 131 refereed scientific papers, and in total there are about 242 relationships that have been assessed. And of those, we were, about 172 were usable. So we had 172 relationships available. Now, many of those were ours that we'd written, but there were studies from Washington, Oregon, Alaska as well that we could dip into. And so here's what they look like. Here's the number of studies. And I think this is an eye-opener for me. A lot of the relationships between something and salmon involve the use of these isotopes of nitrogen, the chemical tracer that has some issues, but it may indicate the presence of salmon. That's not my thing. I don't really care about the chemistry, and it is difficult. There are ways in which you can get false positives. So that's not, I don't think it's nice to have a tracer, but it's not, I didn't want to end there. Disturbance was looked at, nutrient enhancement, food provision, for example, feeding rates, growth. And so these are all at the individual level, individual plants or insects or birds. And then the next level up, you have more of a sort of a community. Well, these are metrics to do with population dynamics. So all those study relationships are available. And then you've got a few that have to do with the community. So community structure, for example, would be, and that would be the plant, riparian plant study that I told you about. So these are all the studies that have been done. The reason we're compiling all these is because we were trying to find out, answer an age-old question is, well, how many salmon do you need to, support, a healthy ecosystem? And this is important in a time when salmon are declining in many parts of their range. And so we found a way to, show how test them all, and it's using the same framework. And so these are different examples of relationships. So, for example, here's the one on the top left is the one I just showed you. We've redone the data. We pulled the data out of all of these studies, and they all have something to do with salmon density on the x-axis and something else. So you can see chum caught per hour by bears. That's an Alaskan study. The more fish there are, the more they're getting eaten by bears. There's one on here showing a change, the drop in invertebrates that happens when the salmon return, and so on. So took all of those studies and said, well, what's the bottom line? How often do we even have a relationship? And what does what do these look like? And here's the answer. In about just over 10% of the studies, there's a straight line relationship where more salmon, the more X. Could be more birds, could be faster growth of fish or whatever. In the other two that you see there in the middle, the green and the orange, you can see that there's approaching some sort of a limit, which is what we often get. So it's going up to some sort of the curve is bending where there's a limit. The more salmon you add, it's starting to slow down. But then a lot of the time you get no relationship at all. And this is interesting. Some of the authors of these studies actually said there was, but when we analyzed their data, we didn't see one. And of course, there's a bias because if people don't get a relationship, they're probably less likely to publish the paper. So the bottom line was that we found 75 positive relationships with number of salmon and some element of the ecosystem, 14 negative ones. So that would be things like scouring out the insects, and sorry, that was sort of the overall pattern here. So for those who say that there's not much evidence that salmon affect ecosystems, well, it turns out that there's about 89 examples as of a couple of years ago that refutes that. But this has a bit of importance nowadays when we think about how we might want to manage salmon, because we don't consider salmon, so we don't consider the terrestrial effects of salmon when we are thinking about catch limits for salmon. So how many salmon do the bears need? How many do the eagles need? I don't expect people to care necessarily about the sculpins in the streams, but it is relevant because, in fact, Canada's wild salmon policy has specified that a challenge is the need for development of an ecosystem objective that's widely appreciated but difficult to quantify. This was an excellent blueprint for the way that Canada was committed through the Department of Fisheries and Oceans to trying to take a more holistic approach to salmon management. But when this was written, it was difficult to quantify because not much my work hadn't been done, and there's some groundbreaking work that had been done by some other scientists, such as Tom Reimken at the University of Victoria, but it wasn't enough to be able to figure out the trade-offs that would be involved in not catching as many fish as you might want to in order to have an effect on the ecosystem. And so it's true, at the time, few studies provide advice on the numbers of salmon necessary for healthy ecosystems. Well, we've spent quite a bit of time working with people in the Department of Fisheries and Oceans, and who are trying to implement this policy. And we now have all the results. Um, but I don't see serious consideration of this anytime soon for the way in which salmon management is going to occur. Um, right now, there is still not an explicit consideration when setting catch limits on on other users of the fish other than human beings. So I think that ecosystem subsidies certainly can be translated into salmon management if people want to do that, but it does depend on decisions that people might want to make about targets and trade-offs. We and others have done the work that shows how they're connecting these ecosystems together, and that in itself has been a wonderful journey of discovery, and we have many more things that we will still want to do on this. But in the meantime, I think we're in a position where if people want to, if they have the will, they can also start to manage the fish in a more holistic way. And I hope that at some point that might become the case. So I should again mention the army of people who've been involved in this research. This is only one cohort of graduate students and postdocs and undergraduates. In fact, there have been many waves of students going through the group all the time, and many of them have never met each other because this work has occurred over a 15-year period. But again, just to emphasize how wonderful it has been working with them. And especially, again, I'm very thankful for my friends in the Celtic community who have taught us so much and helped us keep safe doing the work. And that has been, in some ways, probably the biggest surprise for me when I went in rather naively. I thought I was just going to study fish, but I've ended up really being very, very taken with the whole environment in which the people and the fish together are coexisting. So leave it there, and yeah, happy to chat.
Thank you so much for that wonderful talk, John. I'm very jealous of all those beautiful places you get to go, even though I know you got very wet many times. Um, so now we're moving over to the Q&A portion. So please, for any attendees, if you do have any questions for John, use that Q&A function. Just type in your questions and we'll get to them. To get us started, we have a couple questions though. You talked a little bit about kind of how you got started, but really, what was your main thing that got you interested in this field?
Well, I was a professor in England for 12 years before I came back to Canada when SFU made me an offer I couldn't refuse. And that particular offer was to study salmon. There was funding to allow that to happen. And so I had not actually studied salmon before that. But I was very interested in these wider ecology questions. So I guess what drew me to it was reading some of the early work by people like Tom Reimken at the University of Victoria and others, and I thought this is an excuse to study an awful lot of things at the same time. And so I didn't want to, we in the West Coast didn't need another stock assessment biologist. We have lots of terrific stock assessment biologists. Lots of people look at salmon, from a fisheries point of view. I thought, well, I'm going to look at them from a different angle, and I get to go birding and and botanizing and all of that sort of thing at the same time.
Wonderful, thank you. Okay, another question here. Um, so obviously, you're in the Pacific Northwest. You study the Pacific salmon. But just a question of, is there a big difference between the Pacific and the Atlantic salmon, or are you just specifying kind of where you are?
Yeah, that's a good question. They're quite different because Atlantic salmon don't die en masse at the end of spawning. Depends on the population. There are some which do, but in many cases they will actually go back out to sea, or they will try to. They may or may not make it back for a second or third go, but you don't get those kinds of densities. Now, I should say my perspective is colored by what I know about, say, they're in Europe as well, Atlantic salmon. They're on both sides of the Atlantic. Um, Further north, where the Atlantic salmon populations are healthier, perhaps there would be opportunities to actually have a big nutrient effect. But Atlantic salmon are not doing well right now in the southern part of their range, and so I think we just don't get those kinds of densities that can lead to that. There's been a little bit of work on it, but not much.
Great. Okay, what else do we have here? So it was great to see you celebrating the research of your grad students and postdocs. I agree, and they contribute so much. But did one of them ever come up with a theory that surprised you, or have figure out something that totally subverted what you thought?
Oh, they subvert me all the time. Uh, I'm trying to think one related to. I got to tell you, one of my best moments always as a supervisor is when my students can always drop in anytime, and I like it when they do. And now and then, I've had times when they've said, stop, reading your emails, look at this kind of thing. I love it when they, there's had the occasional student who's practically kicked down the door and gone straight to my whiteboard to draw out something that they discovered. Um, I guess I'm trying to think of a specific example. Um, I think for me, maybe one of the surprises was actually it was with Morgan Hawking, my former postdoc, who led the plant community work. We we were studying a lot of things at the same time. We had, I gave you a flavor of, we had the nitrogen in the leaves, we had the isotope tracers, we had the densities of plants, we had the transects, we had so much going on. And we were kind of worried about how we would break this up into different publishable units. How would you, um, where does the story, one bit start and the other end? How, and it was going to be a lot of work to publish all those papers. And one day, I stood up at my whiteboard and I said, what if? And I sketched out the whole story with him, um, of what the entire everything was all together. I said, why don't we put this into one paper, one mammoth paper? And that's what we did. And I think the result of that was that it meant we could connect it all together in one spot. And I think just sort of seeing that thing materialize in what started off as a routine meeting just to commiserate over how on earth we were going to get through this. And 2 hours later, you could see it. You could see the story. I think that was certainly a pretty good moment.
That's wonderful to hear. I think we just have one more question here. So if anyone else has any more questions, this is the time to get them in. Um, but if not, this would also be a great ending question because this question is kind of what's next for you? Do you have any other big plans or future research or new directions you want to take this in?
So we are continuing to count the fish every fall, but we're not doing as many dedicated specific studies now. And so I sort of cut the research off that I've just told you about up to the point of most of our publications. We still have a number of things that I'm hoping will come out on insects. We've done a lot more insect work like that. And the other thing that is happening is that once I felt I'd answered a lot of the key questions there, I also turned my attention toward island studies and asked about other forms of nutrients that were affecting islands and the number of species on islands. So at the same time that we were kind of, in some ways, winding down the 50 watersheds, we ramped up to a study of 100 islands in the area. And so we were looking at that. And so that work is still coming to fruition. That's kind of ongoing still. And so I guess my answer is really what's mostly next is enlarging the story beyond just salmon now and trying to find out what else controls these ecosystems that we think as being terrestrial. But, I think of them as being islands, in a sea of subsidies. And we're interested in many other relationships like that.
Wonderful. Oh,
Hmm.
great. Me saying that was the last question made a lot more people have questions. So love
Hmm.
to see that. So we have one here. Curious about why plants that don't need the salmon are less abundant where there are salmon. Um, wouldn't we expect no difference?
Yes, it's a good question, Kathleen. And I must admit, this was one, I mean, you could see it in the graph. We could see it in the data. We didn't see it at the time. We didn't know that was going on. Like it doesn't jump out at you, that particular effect. And so the only thing I can think of is that they may be getting shaded out somewhat. So when you get salmonberry can be way over your head, like way over, and you can get these masses of stink current. Like we had to bushwhack through that stuff often, and it's just way over your head. And you know these it's pretty shady under there, so it's possible that that might be driving it. But it is a good question, and I'm not sure I completely understand it. And actually, this brings up one other thing. When we did this research on the Great Plant Survey stuff, this was in 2007, and we hadn't yet started counting the fish in most of those streams yet. So about half the streams, we actually had no idea how many fish were in them. And so that's we were kind of going blind. In a way, I guess it's good. It meant we couldn't be biasing the data subconsciously, but it also meant that it required some hindsight to see some of these relationships between salmon and the riparian vegetation because we didn't know there were a lot of salmon or a very few salmon in some of these streams at the time. Now we kind of get it and we can see it, but I don't want you to think that these results are just immediately necessarily jump out at you. You need a lot of streams to pull out the relationship, and you know, we all know, I mean, I have salmonberry growing as a weed in my front yard. There's no salmon near here. So that, you know, it's not these are not all, these are just sort of overall average effects.
Great. All right. Well, this isn't a question, but it's a lovely comment, so I'm going to read it. Just saying, thank you. Super interesting connections, methods, and discoveries, which I would agree.
Well, thank you. I appreciate that. Yeah.
And then we have another one here. How
Yeah.
would historic salmon densities look on your graphs?
Oh, that's a really good question. Our information on that mostly comes from talking to people in the Celtic community, and there were definitely a lot more salmon in the past. And just in over about the last 20 years, the average number of pink and chum, especially chum salmon coming back to those streams, has dropped by about 50%. So just while I've been studying them, we've seen a decline. It's variable. They bounce around a lot, and it's not. I can't predict one year to the next how many will come back, but it has been going down. So the best information I have for most of those streams comes from people who live there, and there were definitely more salmon. Now, if I were to speculate, then what does that mean in terms of the ecosystem effects? I think there would have been basically more results out of toward the right end of my graphs. I think, you know, where we got this sort of it stopped, like you know, it goes up and it plateaus. The effect on birds, the effect on coho salmon numbers, and various other ones. I think that probably a lot of those other streams would have been further to the right, and it would have they also might have been more maxed out than they are now, except for the ones with the waterfalls and the barriers. Our control streams, they were always controls.
Yeah.
But that's a very good question. I'd love to know. So we now, as I mentioned, we share the data with the Celtic Resource Department, as well as the Department of Fisheries and Oceans, and all three of us count the fish in exactly the same way using standard protocols, so that all of our data are not only being held now by the Celtic nation, but also by the Department of Fisheries and Oceans. And so I'm just saying, I think, you know, we've effectively doubled the number of streams that were counted in that region, and I think whatever happens, we want to keep that going. And so we know a lot more about the actual population numbers quantitatively than we did when we started. But as of course elders and people who with long memories in there, certainly it'd be wonderful if we could somehow fill in more of those blanks.
Yeah. That's wonderful that you have that such a strong relationship with that community and able to get that insight. That's invaluable.
Yeah, I've been really lucky, and they really took me under their wing. I knew nothing when I started that work, and you know, I had just arrived from England, and so I really probably made just about every silly mistake you can make at first, and I was very fortunate to be able to work there and to work with folks as well. So it's been a it's been a real journey.
That's wonderful. Thank you so much. Well, unless we have any other last minute questions come in, we can wrap this up. It's been, I can see it slowly getting darker behind you, John. So the evening is it's coming. So yeah, thank you everyone for joining. It's been such a pleasure to be able to learn a little bit more about your research, and I look forward to seeing um, what else you work on with your wonderful team of students and postdocs. So thank you again, John, for taking the time to share your expertise and knowledge with all of us. And yes, thank you to all our people for joining us. We'll hopefully see many of you at our next Café Scientifique, which will be at the last Tuesday in November. That is all for us here. So look for a follow-up email in your inbox in about an hour or so and have a lovely evening.
Great. Thanks very much.